So now that that's all sorted out, we're going to talk a little bit about how linking phenotype and genotype. Now, what does that mean? So we spent the last few weeks talking about how you can take a microbial community and identify who's in there, right? Maybe a little bit of the relative proportions of who's in there. You can essentially take a census of a microbial population. Not a perfect census. No census is ever perfect, but at least you can get a general idea of who's in there. It's also possible, in fact, it's quite easy, relatively speaking, for a microbial ecologist to look at an environmental sample and see what's going on in terms of the metabolism of that community. And so I give you an example of the nitrogen cycle. And you can do this in a fish tank or a soil sample or whatever. It's easy to measure the concentration of any of these nitrogen compounds. In fish tanks, you buy a little kit, and the tubes turn colors that tell you the concentration of all these things. You can take a soil sample to the extension office just off campus here, and for $1, they'll give you the concentration in that dirt of all these things, plus a bunch of other stuff. Things like water content, sand content, the list of total organic carbon, and all that kind of stuff. So that's easy. It's also easy to measure these arrows. And so, for example, imagine injecting a small amount of N15 ammonium acetate into a soil sample, radioactive ammonium acetate. Pretty quickly, you find ammonium nitrite in that soil because the ammonia gets converted to nitrite. It also shows up in organic carbon pretty quickly. And so it's easy to measure those rates and to follow all these compounds and atoms around through these processes. Of course, if you add organic nitrogen to a sample, it goes primarily to ammonium, and then ultimately it spreads throughout the whole system, right? But you can watch that progress from one thing to another and measure all those rates. You can measure forward and backward rates by adding, for example, radioactive nitrite and measure how quickly it starts going to nitrate, and you can add radioactive nitrate and see how quickly it goes to nitrite. I mean, these things are really easy. The problem is we now have two big piles of information. What's going on in an ecosystem and who's in that ecosystem. But how do you put those things together? How do you find out who's responsible for all these arrows in a microbial community? And usually it's not going to be just one creature, right? It's going to be a collection of creatures that can do any of these steps under certain circumstances. And so tying that information about the phylotype, who's there, and the phenotype, what they're doing, is really hard. There are basically two approaches to this, and we're going to talk about one today and the other on Wednesday. The first approach is to start with the process and then figure out who's doing it. The most common way to do this is called stable isotope probing, which we'll talk about in just a few minutes. The other way to do it is the reverse. You start with who's there, and you find out what they could do. And that's done by using metagenomics. You sequence their genomes and see what genes they have. And we'll talk about that on Wednesday. So let's start with stable isotope probing. Whoops.